you?
Okay, thank you. So hi everybody. So unlike Uri, I'm going to take a different approach and delve into a single project. But before I do that, I want to thank you for the invitation, the organizer, for the invitation to speak here. I am in the Life Sciences Department, so this is right here on the 4th floor. And as you can see, just like Ofer, I really like, well, not Gemini, but DALL-E generated images. So we are on this interface between, ecology and network science, but in data science. So what I want to do in this talk is first I'm going to talk to you about ecology, explain a bit what are the challenges that we have in ecology, and then I'm going to kind of delve into this realm where we try to apply algorithms from data science to understand the ecology. So when we go to the field and we look at this amazing biodiversity that's all around us, this biodiversity is in part of many kinds of ecological interactions. So interactions such as mutualism, here you see plant and pollinator, parasitism, seed dispersal, where animals take seeds and just spread them as they fly, and herbivory. So different kinds of ecological interactions generate, or there are kinds of natural forces that shape ecological communities, bringing them to what we see. But ecological communities and interactions are typically not only pairwise. So it's not only a single herbivore eating a single plant or a single butterfly pollinating a single tree. These come in many networks. There are many species that interact in intricate ways and non-random ways, and we can describe their interactions or the web of interactions using networks. So when I say community in this talk, I'm talking about assemblages of species, and these communities that interact have some structure to their interactions. So that means that this is not random. So I'm not just taking, in nature, we don't just observe species interact randomly, but there is some structure to how these links are distributed across the species. And why does it matter? So here's an example of why it matters. It matters because the structure of these communities or these networks affects the ecosystem services and the stability of these communities to perturbation. So here's an example where we looked at how an invasive species in Patagonia, so an invasive species altered the shape and the structure of these interactions, and this rendered networks that are invaded being less, so more susceptible to perturbations than intact networks. And there are many different kinds of these studies, and this is something that is well known. We look at network structure because the network structure influences the ecosystem services that we get from these communities and the stability of these communities. So for example, even in our microbiome interactions, ecological interactions between plasmids and bacteria shape how our microbiomes and therefore our behavior and our ability to digest food, so on and so forth. Other notable examples are pollination and also disease emergence, such as COVID. All these processes operate on network structures. But we have a problem. The problem that we have is that we cannot get enough information, right? We always have undersampling of our networks. Why does this happen? It happens because, if you are a field ecologist, and I've been a field ecologist, before I became a computational ecologist, I was a field ecologist for many years. And I know, this is like in the bachelor's, so I know that it's extremely difficult to get to the field and look at all these interactions. You need to spend hours in the field, and you miss a lot. So here are some examples. So you set up these covers and you look at animals. You're trying to, make sense of what is happening in the forest. You miss a lot of interactions. This is a bat eating a fruit, but this is from Mexico, but who said we're going to get all the fruit that this bat eats. Here is an example where we actually sample the parasites from animals, but this is hours and hours of work. So there's always, incomplete sampling. And although ecology, just like we told you, has advanced into this era of big data, for example, using methods like ecology or approaches like ecology, but also genomics, where we can, sample a plant, take a plant, look at the DNA of whatever is on the plant, and then say, oh, we find DNA of some pollinator, the pollinator pollinated the plant. This is still not enough. And I just want to give you an example of how, of why. So here's an example taken with a camera trap set up on a tree, again in Patagonia. And you can see we're now documenting interactions between a bird and fruit. So this is bird-fruit interactions. You see, it takes its time, it's eating the fruit. You can't miss it, right? I mean, you could if you didn't put up the camera. But it's very difficult to miss if you have a camera. Okay, on the other hand, let's take a look at this bird. It's here. Okay, so it's here for a second, but it's still not eating anything. Oh, it took something and went away. This is interaction that is very difficult to miss, even if you have a camera, right? Okay, so we have all these methods that I showed you, and we're trying to set up these matrices of interactions where you have pollinators and plants or other kinds of interactions, but you have all these missing links. So what is the problem? Like what are these missing links? So when we look at these matrices, either we see links, so we see these interactions, we observe them, or, which are all these non-white ones, or the white ones, the problem is that we don't know what they are. So maybe they are missing, right? So why are they missing? Because the species I'm trying to sample are very rare, we never see them. We were not enough in the, in the field. Maybe it was raining, maybe I came at night, but the butterfly, is not nocturnal, it's diurnal. Many reasons. So they're missing. On the other hand, some of them are forbidden. So I could spend all my life here, but I would never see this interaction because these organisms, they just don't match. So how do we separate those, and what can we do in order to kind of complete our missing information? So one of the solutions is to predict links using multiple methods. The method that is kind of mostly the most common one is machine learning. So what do? You say, okay, I know. So one approach is to use the traits of the species. So I know the traits of this pollinator. I know the traits of this plant. Using some machine learning model, I will calculate if there is. This is like Tinder, or Cupid, right? You're trying to take traits and match them. The problem is that first of all, you can only do it for a single network at a time, and also the trait data is extremely biased and very difficult to get. So people like birds, so they take, like we said, they take their cell phone and take pictures or whatever. They want to see something, they look at birds, they look at butterflies, they don't look at parasites. So the data is biased. And by the way, biased data is really bad for models, right? All models. Another approach is to say, okay, I don't know anything about the data, but I do know something about the network. So maybe, just by knowing something about how these species interact, I can kind of predict their future interactions. Here the problem is, again, that the data is very sparse, because ecological networks are sparse. Out of, thousands of interactions, we usually only, so out of some potential number of interactions, we usually don't observe more than 10, maybe 20%, and that creates imbalance. So a lot of zeros and few ones, that is not so good for machine learning. So what can we do? So we thought about this problem. This is kind of an emerging topic in ecology. Link prediction has been around in engineering, but no, this is like how Amazon and Netflix sell you stuff, but it's not really common in ecology. So we thought about this and we thought, okay, look, we know something about ecological networks. We know that even though they're very different from each other, so plant-pollinator interactions, host-parasite interactions, even though they're very different from each other, the structures of the networks can be similar. So I can find this structure regardless of what these labels are. We thought maybe we can take advantage of this fact. So together with Professor Rami Puzis from the engineering, from software engineering here at BGU, and this work was led by Barry, who was a master's student in my lab and now is in a PhD. We thought, okay, maybe we can join these data together. We can take networks from different kinds of networks, and in that way increase our data sets, right? And provide better link prediction. So what we did is we took existing data from previous studies. So we have 538 networks. These are the kinds of networks that we have. And these are distributions of some of their basic properties. So we took this data and we saw, first of all, like others before us found exactly the same, that if you throw these networks on a PCA, they're not very different from each other. So it's not like plant pollinator networks are concentrated here, and host parasite networks are concentrated here. No, they're all more or less the same, which allows us to use data from different networks. So we trained a model. What we did is we took this data, we split it into two, into training and test. We trained the model using all the networks, or some of the networks, I will show you, in which we extract network properties, like these ones. This is just an example of network properties that we can use. So we looked at the structure of these networks, and then we trained the model in order to predict interactions in the test set. That's what we did. And with this, after some, this is the kind of the classical machine learning pipeline, we get to this confusion matrix, which I will show you now, to evaluate the model. So when you run a kind of machine learning models, what you get at the end is a bunch of probabilities. So if you got your interaction right, that's a true positive. If you got a zero, if you predicted a zero that was actually a zero, that was a true negative. If you got either of them wrong, then you get either false positive or false negative. And with these, we can evaluate our model. So that's, by the way, basics of all kind of machine learning models. And we compared our model to a model, which is here in orange, to a previous model, this light orange, where they didn't use many networks, they just used one network at a time. And we saw that our model was way more accurate. So the percent of links that we guessed correctly was higher. And the number of ones that we were able to put back was not very different, but our precision was way higher. And this is important because I can just throw ones in all my squares and be 100% correct. So this is kind of lying. So this precision is very, very important and typically overlooked. And we were also able to retrieve more zeros correctly. And when you do this, you usually say, okay, you do these when you say, okay, it's a 50% probability, above 50% probability, that is a link. You can also look at other thresholds. This is a very common ROC curve. If this is a random guess, you see that it doesn't matter what the probability threshold is, we are always above the random guess. Also, another way to look at it is the precision recall trade-off, and here as well, we are above the random guess. But what you can already see here is this fact that some communities, like specifically host parasite communities, provide better predictions than other communities. And this is something that I will show you now. So overall, the overall conclusion is that training our model on multiple networks improves our link prediction. So this crazy idea that we had somehow worked, compared to training on a single network. Essentially what we do is we reduce the false positives in the networks. But as I said, prediction varies by community, and you can see that host parasite networks, this is just two metrics I want to focus on, host parasite networks and plant herbivore networks, plant seed disperser networks are consistently better than in prediction than other kinds of networks. Specifically, plant pollinator networks are not so good for prediction. It's very difficult to predict plant pollinator interactions. Another very cool thing that our model allows us to do, actually really for the first time, is to say, maybe I can predict interactions in one kind of network based on a different kind of network. So what if I train my model on host parasite networks, but then try to predict plant pollinator interactions? Can I do that? So let's see. Each of these panels is a test set, and each of these colors is a training community. So here in the red arrow, for example, is when I trained my model on plant pollinator networks, but tried to predict seed disperser interactions. And you can see that more or less it's the same, but there are some differences. Specifically, when you train a model on host parasite interactions, which is the red, you always get better results, doesn't matter what you test, than when you train a model on plant pollinator interactions. So there is something about these two kinds of networks that makes one of them kind of better predictors and the other worse. I mean, it's interesting to know why, I still don't know why. Okay, so to summarize this, what I showed you is that ecological communities and the networks that they form are crucial for ecosystem functioning, but the problems that we have is that we lack data on their interactions. We have consistent undersampling. That one solution that is, or a solution to this problem is to perform link prediction, but the methods that were until now were very limited, and we came up with this idea of what's called inductive link prediction, where we use multiple kinds of networks, or multiple networks, to predict on other networks. And finally, because we did this kind of crazy thing, that allows us to do this inter-community prediction, and we find that host parasite and seed disperser communities consistently provide better predictions compared to plant pollinator interaction. And maybe that is something that can, with this, we can go forward and generate hypotheses as to why this happens. I have to check this still, but this is ongoing work. Well, so that's it for now. So with that, I want to thank my lab and you. And if you have any questions.
Yes,
yeah, thank you. That was actually very nice. My question is more epistemological. Offer said at the beginning that science, in order to be scientific, one has to use not only inductive methods but also hypothetical deductive methods. And you showed very well the functioning of the inductive method.
Yes.
And I wonder what happened to the second part.
Okay, so the second part. So why do we do this? The reason that we do it is because we want, so when you are a field ecologist, you need to know how to spend your time. So what are the interactions that you are going to want to focus on? This method, or these kinds of methods, they give you probabilities to what are the most likely species to sample. So with this information, right, you can go to the field and focus on what you have missed. That's one. Another thing here is that you really don't have, one of the problems in all these kinds of methods is that we don't have the ground truth. So we need, with this deductive method, with this, what you can do is you can generate hypotheses as to what are the links, like I showed with plant pollinator networks and host parasite networks. So what are the links or parts of the networks that can generate more zeros, more ones, and then go test them in the field, because you really need this, data to kind of go back to the model and validate what this shows you. I hope this answers.
But you do that.
I don't do it, but this is what we do for people to be able to do it. So because my lab is computational and I can do everything. But it came out of necessity, right? Because I spent many years and time in the field, and I know that this is difficult, right? And okay, more questions?
So this is the interaction between computational ecologist and field ecologist. Shai can be a field ecologist as well, but then he would not be promoted because he'll be in the field all the time, trying to find some missing links. Thank you, Shai.